Very good afternoon, students. Myself, Dr. Preeti Devan from JD Patil Samudkar Mahavidyalaya, Darapur. So, we have completed our unit 6, which is revealed with the immunology. Now, we will, from today onwards, we will start our next unit, that is biotechnology and genetic engineering. So, I am sharing a PPT with you. Genetic engineering. So, here you come to know the term recombinant DNA technology. So, in this unit, what we are going to see, we will see this topic. We will define a genetic engineering. Then we will see the outline process of genetic engineering involving some or all the following: isolation, cutting, transformation, introduction of base change, and expression. These are the steps involved in the genetic engineering. Then we will see the application of genetic engineering in plants, in animals, and in the microorganism. So genetic engineering, what does it mean? So definition of genetic engineering: the simple addition, deletion or manipulation of single trait in an organism to create a desired change. See, इसमें क्या होता है, हमको जो यूजफुल कैरेक्टर है उसको निकाल के दूसरे इसमें डालना, that is means addition, simple addition. First of all, we will isolate it, then we will add it in the another organism. Means we are making the recombination of two. That's why it is said to be a recombinant DNA technology also. So genetic engineering, what you need to know, इसके लिए आपको क्या क्या पता चाहिए? So first one, how it get manipulated, manipulation and alteration of the gene. Then process involved, which process are involved? Isolation of the gene. Then isolated gene should be transformed into the another. Transformation means ट्रांसफर करना, उसको एक ऑर्गेनिज्म से दूसरे ऑर्गेनिज्म में ट्रांसफर करना, दैट प्रोसेस इज कॉल्ड एज ट्रांसफॉर्मेशन. एंड एक्सप्रेशन, एक्सप्रेशन मींस हमने अगर उसमें ट्रांसफर किया तो इट विल स्टार्ट इट्स डूइंग formation of the protein that get expressed, that process is called as expression. Then we will see the application. So genetic engineering definition, it is an genetic engineering is an artificially copying a piece of DNA from one organism and joining this copy of DNA into the DNA of another organism. That process is called as genetic engineering. Purpose of genetic engineering. What is the exactly need of genetic engineering? Genetic engineering की need क्यों है? पहले आपने देखा होगा, अभी के भी लोग बोलते हैं कि आपका सब खाना कैसे है, हाइब्रिड है. सो व्हाट डज दिस हाइब्रिड मीन्स? वाइल्ड वैरायटी और हाइब्रिड में क्या फर्क है? तो हाइब्रिड इज प्रिपेयर्ड फ्रॉम द वाइल्ड इटसेल्फ. वाइल्ड में से कुछ कैरेक्टर्स मींस ट्रेट, कुछ जीन्स लिए हैं और उसको नए करके मॉडिफाइड किया है, दैट इज द हाइब्रिड प्रोडक्शन. और जेनेटिकली मॉडिफाइड ऑर्गेनिज्म, और इट मे बी अ ग्रेन आल्सो, जैसे कि राइस के अलग अलग वैरायटीज हमको मिली है. सो व्हाट इज एग्जैक्टली द पर्पस ऑफ जेनेटिक इंजीनियरिंग? द पर्पस इज to allow the genes from one organism to be inserted into the cell of different organism of the different species. हर एक स्पेसीज ऑर्गेनिज्म मींस मतलब वो एनिमल भी हो सकता है और इट मे बी अ प्लांट आल्सो. सो एक जीन एक प्लांट से निकाल के दूसरे प्लांट में डालना जो भी यूजफुल है, दैट प्रोसेस, दैट इज द पर्पस ऑफ जेनेटिक इंजीनियरिंग. उसके वजह से क्या होगा, सभी अच्छे क्वालिटीज जो है एक ही ऑर्गेनिज्म में, एक ही प्लांट में, एक ही एनिमल में आपको दिखाई देंगे. So example, human gene can be inserted into the bacteria. Then human gene can be inserted into the cells from the other animal. Other animal में भी उसको insert कर सकते हैं. Then bacterium gene can be inserted into the plant cell. Means we can insert the gene फ्रॉम ह्यूमन टू द अनदर ऑर्गेनिज्म, और अनदर ऑर्गेनिज्म का भी हम उसमें ह्यूमन बीइंग में ऐड कर सकते हैं. सो जेनेटिक इंजीनियरिंग means that DNA from different organism can be combined. दो ऑर्गेनिज्म के जीन्स जो है वो इंजीनियर करना है. इंजीनियरिंग का मतलब क्या होता है? कंस्ट्रक्ट करना. इंजीनियरिंग हमको किसको बोलते हैं? मोस्टली हम इंजीनियर बोलते हैं सिविल इंजीनियर, मतलब जो कंस्ट्रक्ट करता है. क्या करता है? बिल्डिंग कंस्ट्रक्ट करता है. तो इसमें भी हम क्या कंस्ट्रक्ट कर रहे हैं? न्यू ऑर्गेनिज्म कंस्ट्रक्ट कर रहे हैं बाय टेकिंग द जीन फ्रॉम वन ऑर्गेनिज्म एंड इंसर्टिंग इट इनटू द अनदर वन. So that is the construction. So genetic engineering means DNA from different organism can be combined. So bacterium can be engineered to produce the human protein. So human gene can be inserted into the animal. Overview, we will be say the process of that. The altered DNA is called as genetic recombinant DNA. R हम इसमें R लिखते हैं और R DNA. R stand for the recombinant. So हमको जो नया मिल रहा है दोनों से इंसर्ट करके इससे निकाल के इसमें डाला और जो न्यू फॉर्म हो रहा है DNA, that is called as recombinant DNA. Then the recombinant DNA is joined to the other unrelated DNA in an organism. This is called as gene splicing. Gene splicing मतलब जीन्स के हम टुकड़े कर रहे हैं, हमको हम उसमें से जो यूजफुल जीन्स है वही हम उसको निकाल के आइसोलेट करके दूसरे इसमें डाल रहे हैं. That process is called as gene splicing. Tiny segment of gene are taken out and replaced by the different gene. So what we have seen up till now. So we have seen what is genetic engineering. Then we have seen what is the purpose of genetic engineering. Then we are seeing the three examples of example of genetic engineering. Engineering में कौन से बैक्टीरिया का ह्यूमन में डाल सकते हैं, ह्यूमन का बैक्टीरिया में डाल सकते हैं, और ह्यूमन का बैक्टीरिया का प्लांट्स में भी डाल सकते हैं. That are the example of genetic engineering. So what is the difference between altered and recombinant DNA? What is the meaning of gene splicing? All these points we have seen. Now we will see the transgenic organism. Transgenic organism altered by the genetic engineering. Genetic engineering से जो altered किए हुए organism है that are called as transgenic organism. Transgenic plants भी है और transgenic animals भी है. So genetic material changed by the other than random natural breeding, gene transfer, moving a gene from one organism to another organism. What does the transgenic word means? Trans means crossing from one place to another and genic means the gene. Gene एक place से another इसमें transfer कर रहे हैं, cross कर रहे हैं, that is transgenic. So it means the bit of gene from different living thing have been bolted together. Bolted means stick together and splice into the another organism to make a new one, which does something which the scientists want it to do. अगर हमको कुछ क्वालिटीज पोटैटो प्लांट के चाहिए और कुछ क्वालिटीज टोमेटो प्लांट के चाहिए और दोनों भी क्वालिटीज हमको एक ही इसमें चाहिए, so what we will start to form, we can name it as टोमेटो. have got this. so दोनों की क्वालिटी हम एक करके एक नया फ्रूट बना रहे हैं, वेजिटेबल बना रहे हैं, that is transgenic. So genetically modified, ये यहाँ पे आपको दिख रहा है कलर, अलग अलग कलर की स्ट्रॉबेरीज है. सो व्हाइट स्ट्रॉबेरी कौन से कलर की रहती है? इट इज ऑफ रेड कलर. बट कलर वाला जीन दूसरे फ्रूट से लेके उसमें डाला और ब्लू कलर की स्ट्रॉबेरी फॉर्म की. सो दैट इज अ एग्जांपल ऑफ genetically modified transgenic fruit. So what we have seen up till now, we have seen the word transgenic animal mean. Then what is the genetic organism? Then we have seen the example of strawberry. So stages in genetic engineering. Before that, I am going to share you one video so that we will cover all your concept.
Cut and join genetic material. The process is known as genetic engineering.
So here in this video you come to know what is the exact process of genetic engineering.
It's one of the principles of biotechnology. Now let's explore more about these principles in detail. Most of the principles of biotechnology are based on two engineering branches, that is, genetic engineering and chemical engineering. Genetic engineering is the branch which deals with the modification of the genetic material of an organism. Which genetic materials are we referring to? We know that it's the DNA which is the genetic material for most of the organisms. And what about the remaining ones? Well, in them, it's the RNA which acts as the genetic material, just like in the case of some viruses. So genetic engineering deals with the manipulation of genetic material. Moving ahead with the next principle, which is chemical engineering. The name itself suggests that the principle revolves around the use of chemicals for the growth and the maintenance of cell cultures. For this, the desired eukaryotic or microbial cell cultures are stored in a contamination-free zone. Further, these cell cultures are used for the production of vaccines, antibodies, enzymes, and various other useful products. We will learn about chemical engineering in the upcoming videos. For now, let's just discuss in detail about genetic engineering. Did you know that the first ever genetic modification was done in a bacterium? That's right. And later on, these modifications were applied to animal as well as plant cells. The genetic modification can be done either by adding something new to the existing genetic material, or it can also be done by deleting something from the existing genetic material. DNA obtained after modification is known as recombinant DNA.
What is this recombinant DNA? See, this is a desired gene. We are isolating it and combining with the host one.
Technically, genetic engineering can be done in many ways. Recombinant DNA is one way to pursue it. When genetic engineering is applied to modified organisms, they are known as genetically modified organisms, abbreviated as GMOs. And what organisms can be modified with this technique? Almost all living organisms can be modified with the help of genetic engineering.
It is also called as transgenic animals, or it can be also called as transgenic organism, genetically modified organism. It may be a plant, it may be a bird, it may be a bacteria or virus.
What kind of applications does this serve? Modifications in organisms can give rise to beneficial products. Consider plants, for example. With the help of genetic engineering, we can produce robust crops, pest-resistant varieties, and also...
So these are the application of genetically modified plants. So we can make a pest-resistant plant. फॉर एग्जाम्पल बीटी कॉटन जो है उसमें वो जो कॉटन की वैरायटी है उसमें पेस्ट मतलब जो कीटक रहते हैं उसका अफेक्ट नहीं होता है, सो इट इज रेजिस्टेंट टू दैट पेस्ट.
High yielding varieties of many crops. This was in short about one way in which genetic engineering can be carried out. The other way to perform genetic engineering is by making copies of the genetic material. This is also known as gene cloning.
What is the gene cloning? हम जो है जो डिजायर्ड जीन है उसको निकाल के हम उसको बैक्टीरिया में इंसर्ट करेंगे. बैक्टीरिया इज एसोसिएटेड विद डीएनए वेक्टर. ये टॉपिक हम सेपरेट देखने वाले हैं आगे जाके. वेक्टर इज एन कैरियर डीएनए. सो वो कैरी करके उसको मल्टीप्लाई करता है, एम्प्लीफिकेशन करता है So that process is called as gene cloning. मतलब हमको अगर एखादा प्रोटीन हमको इम्पोर्टेंट है, उसको हमको ज्यादा क्वांटिटी में प्रोडक्शन करना है, so then what we will do, we will isolate it, then we will transfer it into the genetic vector, and vector will amplify that gene and will produce a more protein. So that process is called as gene cloning. Here,
In other words, gene cloning can be defined as the process of making an exact duplicate of the genetic material. But why do we need a duplicate of the genetic material? Let's consider an example to understand this concept. Imagine there's a new product launched by a company in the market. This has some really cool and amazing features which will be of great use to all. Now, is it fine if the company manufactures just one piece? Of course not.
here, for example, the manufacturer of one type of mobile industry will prepare the more copies of that particular product. In the similar way, if we desire one certain product, a protein, we have to make it in the multiple copies. We have to produce a more amount of that protein. For example, insulin.
There will be so many people who will want to buy it. For this, there will be multiple copies of the same product manufactured on a large scale to suffice the demands. This just means that making exact copies of the original product is useful in several ways, isn't it? This same logic applies for genes as well. A few genes which code for certain extreme important proteins, or even the newly modified genes, can be copied several times. This process of making exact copies of genes is called gene cloning. And just as in this case, here as well, clones or copies of genes serve many important applications. This entire process of making,
See here, this is an desired gene, and we are inserting it in the our gene of interest is inserted into the another organism और इसकी कॉपीज बना रहे हैं. तो कॉपीज हम किससे किससे बना सकते हैं? वेक्टर से बना सकते हैं. वन ऑफ द मेथड इज PCR, polymerase chain reaction, that we will see later on.
the copies of the genetic material falls under the umbrella of genetic engineering. So in this video, we learned about the basics of genetic engineering. To learn more, do watch our upcoming videos.
So here up till now we have seen all the basic concept of genetic engineering. So onward we will see the steps of genetic engineering. What we have seen today, we have seen the what is exactly a genetic engineering. Then we have seen the concept of genetic engineering. What is recombinant DNA? What is the application of genetic engineering in day-to-day life? Then we have seen the what is gene cloning exactly. All these points we have seen in the today's lecture. In the next lecture, we will start with the gene cloning enzyme in the recombinant DNA technology. Thank you. Have a nice day.